About a week ago, a new paper came out in the prestigious journal Nature Medicine reporting a pretty strong association between the non-caloric sweetener erythritol and an increased risk of coronary heart disease or stroke. Now, this paper triggered a major storm on social media and also in traditional media, and there was a lot of attention on these findings simply because erythritol has been more and more incorporated as a sugar substitute in many products like energy drinks, energy bars, chewing gums, many other processed foods, that are catering to the low-carb or keto community, to people who aim to reduce their consumption of regular sugar. So in this video, I wanted to give you my 2 cents of what was actually done here, what did the authors find, and what is my take on the study, and is there really a reason for concern, and should people who are consuming a lot of erythritol maybe reduce their consumption or maybe cut it out entirely. Okay, so let's get started with what the authors did and what they actually reported finding. They started with a group of about 1,150 people who came in for a cardiovascular risk assessment. And they simply collected blood from them, and then they followed these folks for about 3 years and observed who did and who did not develop a heart attack or a stroke. Then they went back to the blood samples they had collected, before they had these cardiovascular events, and measured a whole bunch of metabolites, small molecules in the blood of those individuals to figure out if there's any particular metabolite that is predictive of who would and would not develop a heart attack or a stroke. They found a whole range of different metabolites. The top of that list was erythritol. So what this suggested is that people who have a higher concentration of erythritol in their blood would have quite a substantially increased risk of developing a heart attack or a stroke. Now, they recognized that, because they didn't have a hypothesis based on erythritol being a risk factor, this may have just been a chance finding. And so they ventured to repeat this investigation, now specifically focused on erythritol, in 2 additional cohorts, one in the United States and one in Europe. And again, in people at higher risk for cardiovascular disease, they measured the concentration of erythritol in plasma, and then assessed whether there was an association with coronary heart disease and stroke. And in fact, they could reproduce almost entirely, or almost exactly, what they had found in their first cohort. And so they ended up now having basically 3 cohorts in which they had found a pretty compelling association between elevated levels of erythritol in blood and an increased risk for coronary heart disease and stroke. Now, obviously, that by itself isn't very meaningful. It's just an association. There isn't, really any good evidence that there's a cause and effect relationship underlying this association. So they conducted a few additional experiments. One of these experiments was to actually investigate potential mechanism through which elevated concentrations of erythritol in plasma could increase the risk of coronary heart disease or stroke. And what they did is they conducted a whole bunch of experiments in a test tube and also in mice, in which they added different concentrations of erythritol to platelets, the type of cell in blood that is very much involved in blood clotting and formation of thrombuses that actually is crucial in the events of both coronary heart disease and stroke. What they found was that once a certain threshold was crossed, that higher levels of erythritol actually did increase the ability of platelets to clot, and also thrombuses formed much more quickly in mice. So yes, basically erythritol here in these test tube experiments and in mice was found at elevated concentrations to actually increase the risk of blood clotting. And this was then presented as basically the potential mechanism through which elevated erythritol concentrations in blood may increase the risk of having a cardiovascular event. Obviously now, because erythritol is a component of food, this triggered the question, could it be that the elevated concentrations of erythritol in blood are actually related to consuming more erythritol in the diet. Now, for these three cohorts in which they established that there's an association between blood erythritol and cardiovascular events, they did not have any dietary data, or at least they didn't present them. So they conducted a separate experiment in which they recruited just 8 healthy young people and gave them a single dose of erythritol, 30 grams of erythritol in water, asked them to drink this, and then they just measured erythritol levels in blood repeatedly over about a week. So it's worth looking a little bit more closely at this experiment. So they started with measuring the blood erythritol level in these 8 healthy people. And the first thing to note is their blood erythritol level was in a range of about 3 to 5 micromolar. Then they gave them a dose of 30 grams of erythritol, and that dose was based on the fact that the top consumers frequently consume about 30 grams of erythritol per day. So they administered the beverage containing 30 grams of erythritol, and within 30 minutes, the level shot up about 1,000-fold on average, reaching levels between 3,000 and 5,000 micromolar. Stayed elevated somewhere in the range of 1,000 to 3,000 micromolar, and then came down roughly to baseline at day 3 or so. So I saw a number of people share on social media that they felt this was pretty conclusive evidence that dietary erythritol leads to elevated blood erythritol levels, and that would be increasing the risk of heart disease and stroke. And they thought, this is enough evidence for them. They're not going to eat erythritol ever again. Now, I do not share this interpretation of these findings, as I have a number of concerns, and I'd just like to go through these concerns with you. So my first concern is what we would call a lack of generalizability. Now, in this particular study, they studied the relationship between erythritol levels in blood and cardiovascular events in 3 different cohorts, including, I think, total was something like 4,000 or so participants. But these were mostly pretty sick people, fairly, elderly, mid-60s to mid-70s in terms of the age. About half or so had already had a heart attack. About 20% or so suffered from type 2 diabetes. 3/4 had hypertension. So this was not your average, normal, healthy population. And so what this really means is that if you're in your 20s or your 30s or your 40s, or even older, but you haven't had a heart attack yet, you don't have diabetes, you don't have metabolic disease, it's really unclear whether or not these findings are at all relevant for you. So lack of generalizability is, in my opinion, a major potential qualifier that we should keep in mind here. My second question or concern is whether blood erythritol actually is a good biomarker for dietary erythritol. And this is based on the fact that our bodies can actually make erythritol ourselves. There's a pathway called the pentose phosphate pathway in which glucose can be converted into erythritol. So whatever erythritol we're measuring in blood could be derived from dietary erythritol directly, or it could actually come from dietary glucose that gets converted into erythritol in our body. And this is linked to my third concern, and that is that there's a whole bunch of factors that previous publications had shown are associated with an increased level of erythritol in blood. Specifically, people that are older, that weigh more, have a higher fat mass, or that suffer from obesity, people who have type 2 diabetes, or people who have chronic kidney disease, so CKD, they have a higher rate of conversion of glucose through the pentose phosphate pathway to erythritol in our body. And so they tend to have increased levels of erythritol in blood. And so, in other words, if we're having someone who has elevated levels of erythritol in their blood, it could be because they ate a lot of erythritol recently, or it could be because they have a major metabolic disease, or because they're older, or maybe because they're obese, or maybe all of these, right? Now, this paper actually does provide some clues as to whether the erythritol measured in blood was largely derived from diet or from endogenous synthesis. As we saw earlier, the baseline blood erythritol level in healthy people before they consume erythritol was about 3 to 5 micromolar. And so if now we were to add the concentrations measured in the blood in the different cohorts in this study, it would look roughly like this here. What we're seeing is whether people develop a cardiovascular event or not, there's a very wide range, roughly from about 5 micromolar to about 5,000 micromolar, with on average maybe slightly higher blood erythritol levels in those who go on to develop a cardiovascular event. But what we note is that basically in all of the roughly 4,000 people that were studied here in these different cohorts, the blood erythritol level is substantially higher than what we would expect in healthy people who are not consuming erythritol. So this can mean two things. It can mean that basically every one of these people regularly consumes erythritol. And if you ask me, that's extremely unlikely. Or to a significant degree, the blood erythritol levels here are elevated because the people studied here are not healthy people. They are people who are sick, who have diabetes, kidney disease, heart disease, who are obese, and who are older. All conditions associated to some degree with an upregulation of erythritol synthesis from glucose in the pentose phosphate pathway. Another piece of evidence that supports this is that the distribution of blood erythritol levels in the different cohorts in the United States and Europe were almost identical, even though estimates of erythritol consumption are probably 2 to 3-fold higher in the United States than in Europe. So that doesn't rule out that some people may have elevated erythritol levels in blood because they regularly consume erythritol in their diet, or at least they consumed erythritol the day before this blood was drawn. But to me, it's actually very strongly suggestive of the idea that most of these individuals have substantially elevated erythritol levels because they are sick already. Now note again, this on the y-axis, this is a log scale, so a little bit higher could actually mean 10-fold higher than the baseline we would expect in healthy people. So keep this in mind. Now, the particular concern here is that all of those factors, higher age, obesity, type 2 diabetes, chronic kidney disease, they're also all major risk factors for coronary heart disease or stroke. And so this is a concern because what may be going on here is what we call confounding. So when we're looking at a observational study like this, and we're finding an association between an exposure, in this case erythritol levels in blood, and an endpoint, in this case the risk of heart disease or stroke, what we're really hoping for is that this association kind of is reflective of a cause-effect relationship, right? That the higher levels of erythritol in blood in some people are actually the cause of these people having a heart attack or a stroke. Now, if we have an issue like this, we can never be certain of this, by the way, in an observational study. It's just that this is the hope why we're even doing these types of studies, that we're trying to get at these cause-effect relationships. Now, a major problem in us establishing whether there's whether there could be this type of cause-effect relationship is that there could be this issue of confounding. Confounding simply means confusion of effects, right? So what we're thinking is there could be an effect from erythritol in blood to cardiovascular disease, but maybe we're confusing this. Maybe what's really going on here is that those people who have an elevated level of erythritol in blood are just those same people that actually have metabolic disease. They're older, they have type 2 diabetes, they have chronic kidney disease, and that is actually why they're having a higher risk of having a heart attack or a stroke. And maybe erythritol has nothing to do with the higher risk of heart disease or stroke. Now, in an observational study, we can try to address this, and the authors did this in this study by adjusting for these factors. So they did adjust for age, they did adjust for a measure of adiposity, and they did adjust for whether or not people had type 2 diabetes. But they didn't adjust for chronic kidney disease. They just ran some separate analyses there, which they stratified by kidney function, which is not exactly the same. But I think it's fair to say that there's some potential here for what we would call residual confounding. So it's very well possible that even though they adjusted for some of these factors, that it's still that the relationship that is of interest here between blood erythritol and an increased risk for a cardiovascular event is confounded by the fact that people with higher erythritol could simply be those who are sicker, older, more overweight, and so forth. And then my fourth concern is the risk of confounding by dietary factors. Let me explain this here. So if we're really interested in whether increased consumption of erythritol in our diet could increase blood erythritol, and that could in turn increase the risk for coronary heart disease or stroke, we need to be conscious of the fact that people who eat the most erythritol may have very different diets in many other ways. And I just listed a few of these factors here. So the people who consume the most erythritol in their diet may be those who follow a low-carb or keto diet, and they may have, for example, a higher total fat intake, higher intake of animal fats or saturated fatty acids. They may have a higher intake of, potentially ultra-processed foods, simply because erythritol is added largely to foods that probably fall into the category of ultra-processed foods. They may have a lower fiber intake simply because, if they avoid carbohydrate-rich foods, they may be getting less fiber. So a whole bunch of factors there, and it's certainly possible that one or another of these factors could affect the risk of coronary heart disease or stroke. Now, you may say, well, in this case, they didn't even have data on dietary erythritol. I say it doesn't matter, because if in this study they measured blood erythritol with the thinking that this may be at least partly reflective of dietary erythritol intake, then it's still possible that these other dietary factors, whether they were measured or not, could be confounding the relationship of interest here, right? And so it could very well be, again, that people with higher blood erythritol levels simply have a higher risk of coronary heart disease or stroke, because there may be people who have a totally different diet than people who have low blood erythritol levels. At least that's a possibility that these investigators were not able to address simply because they didn't have any dietary intake data, and so they couldn't adjust for those. So I do think this is certainly a concern. So my fifth and last concern is whether those tests that they use to assess a mechanism here, in which they showed that in a test tube or in mice, erythritol increases the risk of blood clotting, whether this is actually representative of what would happen if erythritol levels were elevated in humans. It's quite often that experiments work out quite nicely in a test tube, but then they can't be reproduced in humans. So we should be really skeptical of this. Now, there's another reason to be skeptical here, particularly, and that is related to the little kind of human study that they did in which they exposed people to erythritol. As I mentioned, they gave 8 people 30 grams of erythritol simply to measure the degree to which plasma erythritol levels would change in response to this dietary erythritol exposure. They registered this as a clinical study on clinicaltrials.gov. So this is publicly available. You can take a look at this as well. And here you can actually see that they, in the pre-registration, they said that they would enroll 40 participants. Now, 40 participants is quite a bit more than 8. So they said they're going to study 40 people, but they reported data only on 8. Now, it's possible they only studied these 8, but it is a cause for concern that maybe they studied more and only reported data from these 8. Now, the second concern is that they defined a priori, so before they actually did the study, they defined their primary outcome measures. And so the primary outcome measure here was platelet aggregation after polyol ingestion. So polyol is just another term for sugar alcohol, and they studied 2 different ones, xylitol and erythritol. And so they wanted to actually check in these 40 individuals whether exposure to 30 grams of erythritol changes platelet aggregation in vivo in these individuals. Now, none of these results is actually shared in the paper, and this raises a major concern, and that is that the investigators may have done the work, maybe in 40 people, maybe just in 8, but they almost certainly also looked at whether 30 grams of erythritol changed, platelet function, and specifically, aggregation and coagulation. And the fact that they didn't share this in the paper suggests that they didn't find the result they were looking for. So to me, this is a major red flag, that if you have a pre-registered study in which you quite obviously wanted to provide some or generate some additional information that would support the overall story that you're trying to tell with your paper, and then you're only sharing some of those results from that pre-registered study in only some of the people, it makes me wonder about what they actually found or did not find in this study. So what do we conclude from this? Is this really conclusive evidence that dietary erythritol intake is a risk factor for coronary heart disease or stroke, as some people have suggested? I don't think so. I personally don't think this is conclusive. It's really not definitive in any way. I do think it's possible that erythritol actually is causally related to cardiovascular events, possibly even through an increased risk of blood clotting. It's very well possible also that erythritol in blood is simply an innocent bystander, that is a factor that is elevated in people who are generally sick, who have a lot of conditions already that are risk factors for coronary heart disease or stroke. Maybe people who are old, people who are overweight or obese, people who have type 2 diabetes or who have kidney disease or other conditions that are associated with metabolic disease and oxidative stress. And in those people, there may be more conversion of glucose through the pentose phosphate pathway, leading to higher plasma erythritol levels, but maybe the erythritol doesn't do anything. Maybe the real story here is that people who are more sick have a higher risk of having more sickness in the form of heart disease and stroke. So I'd say both of these possibilities are there, that there could be something to this, but it's really not clear. In my mind, this is not conclusive at all. And what we really need are, studies in which we look at actual dietary intake of erythritol and look at whether that is associated with cardiovascular event risk. In which studies we could also assess other dietary variables and adjust for those, adjust for the potential of confounding by other dietary variables. And I think it would be helpful to have randomized controlled trials in people who regularly consume erythritol or not, to see whether there's a difference in blood clotting and coagulation in general. So I think these types of studies would add additional puzzle pieces to increase our certainty or that there is potentially a link here between erythritol in the diet and an increased risk for cardiovascular disease and specifically heart disease and stroke. This is a bit frustrating because, who wants to hear this, that now we've done a study and we have either this result or that, and, we don't really know for sure yet. I understand it's frustrating, but I think personally that's the state of where the science is in this field right now. So I think my best suggestion is for you to evaluate, do you need to continue consuming these products, or are there maybe other alternatives with other, non-sugar sweeteners that you could instead use, until we have better evidence on whether or not there is an actual cause and effect relationship underlying the association seen in this study. All right, hope this was helpful for you, and until next time, bye-bye.